So welcome to aerobic respiration videos. So today we'll be discussing only stage 1, which is glycolysis. If anyone doesn't know what aerobic respiration is, it's basically consumption of oxygen to produce ATP. Right. Aerobic respiration level is A-level level. We'll just keep it in A-level level. Aerobic respiration stage 1, right? Glycolysis. So whenever you see lysis in biology, it means breakdown, such as with the photolysis, hydrolysis. Glyco, let's just say it's glucose. It could also mean glycogen, but it's just glucose here, because glycogenolysis means actually breakdown of glycogen, but glycolysis means breakdown of glucose, right? It could also mean glucolysis, I'm not sure, but it's just glycolysis. So let's do it. So what happens in glycolysis is a glucose molecule is phosphorylated. So phosphorylation is addition of phosphate group. So a phosphate group is added to a substrate or any reactant molecules to make them more reactive. Yeah. So phosphorylation here by 2 ATP molecules. So 2 ATP molecules are actually hydrolyzed here, and the 2 phosphate groups are added to the glucose to make it more reactive and to form exose bisphosphate. So it's a 6-carbon molecule. Oh no. Yeah. You see here by, it means 2 phosphate. So exose means exose sugar, 6 carbon. 6 carbon, second phosphate. 6 carbon, 2 phosphate. So it has 2 phosphates attached to it, which makes it more reactive. And it's very unstable, by the way. So it actually splits down into triphosphate. 2 triphosphate molecules, actually. And these triphosphate molecules are actually oxidized to form pyruvate. Right? Actually, to form pyruvate. It's a 3-carbon molecule. Once again, so the triphosphate molecule here are oxidized to form pyruvate. So, by the way, oxidation, you have to remember the old oil rig. It's very good oil, actually. We used to use them since GCSEs. So oxidation is a loss of electron. Reduction is a gain of electron. So basically, in oxidation, there's also a loss of proton. A proton is lost. And in reduction, a proton is gained, and also an electron. It's all big process, and you come across it a lot of times in biochemistry. I'm pretty sure in every single process there is either oxidation or reduction. So to form pyruvate molecules, 3-carbon molecules, and in this process, when the oxidation happens, as we know, the triphosphate loses electron and a proton. Right? When this electron and proton are lost, something needs to gain those. That is how the biochemistry works. Something needs to gain them, the lost hydrogen ions and electrons. If not, the whole process does not continue, right? So the electrons and hydrogen ions binds to NAD, reacts with NAD. NAD, by the way, is a coenzyme. And it can also be said as nicotinamide. I don't know if I remember it. Dinucleotide. Yep, exactly. In light-dependent reaction and light-independent reaction of photosynthesis, we know NADP It's exactly the same nicotinamide dinucleotide, but it is phosphorylated, so it has a phosphate group in photosynthesis. So that is why it's called NADP. But here, it does not have a phosphate group, so it's called nicotinamide dinucleotide, just NAD. It's a coenzyme. So the lost electrons and hydrogen ions from triose phosphate are gained by NAD, same in the here as well, to form NADH. So NAD is reduced by these lost electrons and hydrogen to form NADH. And there is a bit of substrate level phosphorylation here. These phosphate groups are actually lost and added to ADP molecules to form 2 ATP molecules on the both side. So it's total 4 ATP that are gained. But we already used 2 ATP molecules here. So the net gain is only 2 ATP molecules from the process of glycolysis. Right? So that is the whole process of glycolysis. And by the way, the diagram looks so stupid, like it's a bit messy. Yeah, I'll probably try to paste a nice diagram next to the video. So thanks for watching.